Thank you all for joining us today. So just in case there's anyone here who's unaware, we do have an internal website for our BC associates. The link is listed here. It's also present in sort of the emails that I've been sending out to you that have the newsletter. And just to give you an idea of sort of the member benefits, we have monthly newsletters that highlight different research and different events that are happening at BC. One of those events is always our monthly speaker series where we feature a different speaker every month and we give an extended presentation of their research. Also posted on the website are past presentations that have been we've been given permission to share. And we also list our different social activities, for example, book clubs, World Bee Day, lunch, potlucks, gardening, outreach, etc. One of those events coming up is our winter social. That's coming up in 2 weeks from now, so we hope to see you all there. Just some logistics for the presentation today. Please remember to mute your microphone during the presentation. You can also go camera off if you would like. Please also save your questions until the end of the presentation. You can list them in the chat or in the Q&A, or yeah, you can list them in the chat if you would like. This meeting today, it will be recorded and it will be posted to our YouTube channel after the results are published or when our speaker says that it's okay to publish. So this one will be available later on. We recognize that many Indigenous nations have long-standing relationships with the territories upon which York University campuses are located that precede the establishment of York University. York University acknowledges its presence on the traditional territory of many Indigenous nations. The area known as Takaranto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, the Huron-Wendat, and the Métis. It is now home to many Indigenous peoples. We acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. If you're interested in learning more about the sort of different traditional territories of Indigenous people through the world, you can visit native-land.ca and check out different areas, perhaps where your specimens come from, areas that you've traveled to, areas where your research takes place, etc. And with that, I would love to hand over the presentation to Dr. Makaylee Crone. Makaylee is a postdoctoral fellow in the Rehan lab who studies wild bee nutritional ecology. They completed their PhD in ecology at Penn State in 2023 and currently study the microbiome network of bee-plant interactions in urban spaces. Today, their presentation will be titled Nutritional Ecology Across Bee Species. And so I'd like to now, yes, turn it over to Makaylee.
All right, thank you. All right, so thank you everybody so much for coming today. I'm really excited to share some of the work I conducted at Penn State, where I studied nutritional ecology of social and solitary bee species. So we know that both pathogens, pesticides, and landscapes can be really harmful in a leading cause of bee decline. I should say poor landscapes. But nutrition or poor nutrition is also one of the leading causes of bee decline. And so it's very important to study, I think. And it's also interesting because poor nutrition can exacerbate the effects of other stressors such as pesticide exposure, parasites, and pathogens. I do want to go through a little bit of the basics before we sort of get into the studies to kind of orient you. So bees obtain their nutrients from pollen and nectar from flowering plants. Pollen provides proteins and lipids, and nectar provides carbohydrates and micronutrients. We do know that plant families can vary greatly in their protein to lipid ratios. So here I'm showing a study from our research group at Penn State. You can see along the x-axis, we have the mean protein to lipid ratio, and on the y, we have different plant families. You can see that they range from very protein rich towards 0 to 1 to very lipid rich, around 8 to 1. And so because we know all this, this gives us sort of a wide open space to conduct this type of research. There's so much we don't know, but I do want to talk about what we should consider when investigating a species' dietary preferences before we get into the studies. So there are multiple components of a bee species' dietary preferences. First, you would want to look for a nutritional target or an ideal ratio of those nutrients that the bees need. You also want to look at the niche breadth surrounding that ratio, which is sort of the tolerance range a species can tolerate, I guess, and the foraging behavior that a species utilizes to meet their nutritional needs. So to go into that a little bit more detail, a nutritional target or that ideal ratio of nutrients, we generally think about that with the geometric framework for nutrition, so we can sort of visualize that. Here I'm showing an example with proteins and lipids on the axes. You could have any two nutrients on these axes, but this is what's applicable to my research. So you can sort of imagine this as a plane where we have all of these different diets or flowers that bees have access to. So you might have a high fat diet and a high protein diet in this environment. But the bees' ideal intake ratio will fall somewhere between these two diets. So they would have to sort of mix and match as they're foraging to reach this ideal ratio. To talk more about the tolerance range, here I'm showing sort of again this axis with different protein to lipid ratios. And I'll show a few different species and kind of talk about generalists versus specialists. So here we have species A and species B. The niche breadth would be considered what falls between the ends of these two parabola. And then you would also have area where a niche breadth would overlap between species, where they might experience competition. So we would consider species A and species B probably to be quite generalist, and that would result in them probably competing for resources where this overlaps, which is sort of one of the downfalls of, or a trade-off of being a generalist. Of course, we can only really say generalists versus specialists when you compare multiple species. And so species C would be a great example of specialist. It has a much narrower range, and the trade-off for being a specialist would be you don't have access to as many resources, but you're not competing as much for those resources. And finally, just the foraging behavior that the species would utilize. So which plants are they visiting in the landscape? How are they foraging? Things like that. And like I said before, understanding the nutritional needs of multiple bee species is great because it will aid in conservation strategies, such as just plain habitat restoration, but also agricultural supplementation to keep bees in the area and keep them healthy when crops are not in bloom. Okay, so this is the first study, pollen protein and lipid content influence resilience to insecticides in honeybees. And this study was sort of inspired by this paper by Schmiel et al. In 2014, where they took day-old honeybees and they put them into these cup cages and they fed them different diets while they exposed them to pesticides. So here along the bottom is the day that's days past, and then this is the cumulative survival. And each of these lines would be the different treatment groups. So we have a long-term pollen diet where they were fed pollen for the entirety of the experiment, a short-term pollen diet, and a long-term protein replacement to sort of mimic the pollen, and sucrose alone. And you can see that The long-term pollen diet performed significantly better than any of these other groups, which sort of suggests that it might be the lipids in this pollen that are most important, since a protein source didn't really help the bees, though we know now that this is much more complicated in terms of all the different things that are in pollen. But at the time, we were mainly comparing proteins and lipids. Okay, so the study objective was, does pollen protein to lipid ratio improve resilience to honeybees exposed to pesticides? And I tackled this question by feeding bees altered diets where I added proteins or lipids to a base diet and conducting the same survival analysis. I also did this with natural diets, so just different honeybee pollens that we had collected in the field. I also looked at gene expression of a few key genes when they were fed these diets, and I also looked at levels of deformed wing virus. And my hypotheses were that bees fed lipid-rich pollen would be more resilient to pesticide stress and have lower levels of deformed wing virus than bees fed protein-rich pollen. And I also hypothesized that bees fed different diets would have differentially expressed genes. So here are the methods. I had 5 different colonies this first year that I conducted this experiment with about 2,100 individual bees. I would take emerging brood from a colony, and as they emerged, I would place them into these cup cages on day 1. All of the bees would have different protein diets in these little patties down here. And from days 1 to 4, every cage received unaltered sucrose solution. On day 5, the treatment group started receiving a chlorpyrifos sucrose solution that they received every day until the end of the study. And so the different diets I tested, I had a control which received no pesticides, an unaltered pollen diet that all of these did receive pesticides. This is a very lipid-rich diet, a sort of mid-range protein diet, and a very protein-rich diet. And I altered this pollen by taking a base honeybee pollen, grinding it up in a coffee grinder, and then adding canola oil to make lipid-rich pollen, or casein to make a protein-rich pollen. So here are the results from the altered diets. The little skull and crossbones denotes that those bees did receive pesticides throughout the experiment. So at the top here, which performed significantly different than all others, unsurprisingly, is the control diet. And then we have all the different sort of bees that were treated with pesticides. So the 5.5 to 1, remember that's that unaltered diet that was just regular pollen, and the very lipid-rich 1.5 to 1 performed significantly better than these other groups, which included the more protein-rich pollens and the sucrose diet alone. So we concluded that bees fed diets between 1.5 to 1 and 5.5 to 1 have longer survival times than those fed high protein diets when they're exposed to pesticides. But again, this isn't exactly realistic. So we wanted to use diets that we had collected in the field. We used these pollen traps on the front of honeybee colonies to collect pollen throughout the year. And so again, here I have my different treatment groups ranging from very lipid rich to very protein rich, and then again, a controlled diet. So when looking at these results, these are a little bit more complicated, which you would expect given that they're more realistic. So again, this control group is performing significantly better than all of the other groups. But then these diets from 1.3 to 1 to 8.4 to 1, well, I should say 1.3 to 1 and 3.2 to 1 are performing significantly better than the very high protein diet. But these other diets sort of in the middle did not. So it's a little bit more nuanced when you have these natural, real diets that don't have additives in them. So we concluded that bees fed natural diets within this range had longer survival times than those fed a high protein diet. And then when I looked at DEGs and DWV, I found that these genes, all of which I targeted with qPCR, I didn't do genome-wide studies or anything like that. I found that 2 of the detox genes were upregulated, a gene involved in the immune system upregulated, as well as VG, which is just a general gene that's generally upregulated with good health and good nutrition. And then a gene involved in the deoxygenation of free radicals. And then unaffected by diet were another detoxification gene I tested for, the toll pathway, which is involved in immune defense, and DWV levels. We did find different levels across the different colonies, which is why for the second experiment, we included 6 different colonies, but we didn't find that these levels were driven at all by dietary treatment group. So what's going on here? Why might this be the case? So what I think is happening is that increased lipid metabolism is playing a role in pesticide detoxification. I found a paper by Rand et al. In 2015 where they exposed bees to nicotine and they observed increased lipid metabolism. And nicotine tends to target neurons in the brain similarly to chlorpyrifos, which is a acetylcholine esterase inhibitor. So these work somewhat similarly. They're not targeting the same receptors, but I thought this could be a possible explanation for why this is happening, though, of course, we would need to do further, more targeted experiments to see if this is the case. And also, it could just be that bees have a target diet that they must reach, and the tolerance range isn't as wide, and, you know, perhaps they're just dying more quickly if they're not receiving a proper pollen diet. We did put all of our data into a model, and the best predicted diet was 2.8 to 1. I think that this could be the case, but it's less likely just because all of those natural diets that span the range from 1.3 to 1 to almost 13 to 1 that the bees collected themselves over a few days, that's a very wide range. So you would assume if the bees are collecting this, then the tolerance range wouldn't be so narrow. But again, we're not certain. More testing would need to be completed to know for sure. Okay, getting into the next study, more than mesolectic, characterizing the nutritional niche of Osmia cornifrons. So while honeybees are great and we love them, 90% of the bees in the world are solitary species. And to sort of get at the tolerance ranges of the solitary species, I chose to investigate Osmia cornifrons, or the horned mason bee. This is a great sort of gateway bee to sort of do these types of experiments with, because they are used in agriculture, particularly in fruit production in places like Pennsylvania. So they can be produced at a large scale. But again, also they're in the family Makayleedae, so they're different from Apis and Bombus in family Apidae. So just to talk about the mason bee seasonal life cycle, because this is a bit different from the honeybee, in the spring, early spring, these bees will emerge and mate before mom will go and find a cavity to nest in, which she'll provision with pollen and nectar, forming a pollen ball. This is an example of sort of what you might see in somebody's backyard. Of course, on a commercial scale, it's much different. You have these giant mail tote boxes filled with all of these cardboard tubes, generally with chicken wire on the outside to prevent bird and mouse predation. But this is just an example. And each of these individual cells that mom provisions are separated by mud, which is sort of why they're called mason bees. And each of these pollen provisions have an individual egg laid on top. Throughout the spring and summer, the bees will eat the pollen provision, develop into larvae, and they will eventually spin a cocoon before developing into pupa and overwintering to emerge the following spring. And so Those three things I talked about earlier in terms of what do we need to know when we're looking into what makes a bee's diet. It doesn't always start at the target ratio or the tolerance range. Oftentimes it starts from simply recording what the bees happen to visit. So we do know that these bees visit Rosaceae and Fabaceae plants, those families, and they are considered mesolectic because they're not quite specialist enough to visit an individual family or species, but not quite generalist either. In the paper I mentioned before, where I showed all of the different plant family protein to lipid ratios, we also did look at the target ratios of a few different species, including Osmia cornifrons. And we found a mean target ratio of about 2.9 to 1, and the tolerance range shifted from about 2 to 1 to 3.7 to 1. So each of these are individual pollen provisions that I looked at. Something I do want to note, though, is that there were only 10 samples in this study, and the range in which we collected them was not very large. So I really thought that this merited further investigation, and I wasn't quite ready to conclude that this is truly the target ratio and tolerance range of these bees. But that does give us a pretty good place to start. Again, when you look at this chart, you can see that Fabaceae and Rosaceae, those preferred families, have protein to lipid ratios of 1.6 to 1 and 3.8 to 1. So you would imagine that their tolerance range would probably fall between these two extremes. So this brings us to the research questions I addressed. First, what is the protein to lipid ratio nutritional target in niche breath, and how do concentrations vary across sites at time? Because I did want to see if there was sort of a location and across foraging season effect. Secondly, do Osmia collect pollen from a diversity of plants during each foraging trip? So are they collecting different plants within one foraging trip and sort of balancing these different pollens as they go, or are they balancing over time? Perhaps as a result of simply collecting within a certain range of the plants they prefer. And finally, do protein to lipid ratios found in the field correspond to the optimal diets for Osmia during development? So we wanted to see if the tolerance range was perhaps wider than what mom was actually collecting in the field. So we did some in vitro rearing experiments to look into this. So starting with this question first, I went to the Penn State Fruit Research and Extension Center in southern Pennsylvania. I had 4 different sites that would be considered independent when you consider how widely Osmia cornifrons forages, and I collected 10 pollen provisions from each at the end of the field season. And to look at proteins, I used a colorimetric Bradford assay to get the concentration, and to look at lipid concentration, I used a sulfophosphovanillin assay. So I'm going to show a few graphs with our results over time across these sites. Sites is along the bottom, and the concentration of the nutrient is along the y-axis. But we didn't see any significant differences between protein concentration, lipid concentration, or protein to lipid ratio across these different sites. But we did notice that there was quite a broad range in protein to lipid ratio. I can go back and show from around 1.1, 1 to 1 all the way up to 5 to 1. So it's a pretty big range for a single species. And then when I went to compare over time, I used the Penn State Arboretum as a rearing site, and I used trap nests to have the bees lay pollen provisions throughout the spring that I would collect each week throughout May and June of 2019. And there were 10 samples collected each week. So these results are a little bit more interesting, I'll say. We did see differences in protein concentration between weeks 1 and 3, as well as differences in lipid concentration between weeks 5 and 6, and then multiple differences between weeks for protein to lipid ratio. And the overall mean we found across all of these different samples was 3 to 1, median was 2.9 to 1, and the range was astounding. It was from 0.75 to 1 to 7 to 1, which I consider huge. So what's interesting too is that these values are very close to that initial subsampling we did, but the range is nowhere near as close. And what we think might be happening is that macronutrient content is varying over time due to shifting plant communities, because when these bees emerge in early May, generally the only thing out is something like apple. And then apple goes out of season. And you have all these different fruit trees that come into season that the bees are used to pollinate. But because I didn't record which plants were in bloom during certain times, across the weeks in the Arboretum, I can't truly say if, these differences are due to different plants going in and out of bloom. But I think this is more likely than, say, the bees needing a different ratio between weeks 1 versus weeks 4. So this is what might be happening. Secondly, do osmia collect pollen from a diversity of plants during each foraging trip? So to look at this, I went to the Pennsylvania State Game Lands and I collected both nests that had fully provisioned cells, as well as individual foragers that had single pollen loads on them from one foraging trip. And I collected these across 3 weeks. I then used DNA metabarcoding to identify the pollen that was on these bees. I do want to thank and acknowledge these folks for their help with this part of the project. They taught me so much, and I'm really appreciative of them. So first, we would start by washing the bee to remove the pollen. We would conduct a DNA extraction on those samples. Then we would use PCR to target regions ITS1 and ITS2, which are ribosomal regions in plants that would allow us to determine which plant each of these pollen grains came from. We would then run a gel conformation, submit this data for sequencing, and then I would conduct an analysis using these custom Python scripts in curated databases, primarily using vSearch as an algorithm. Okay, so these are the results. You can see that along the bottom we have the number of genera, so the genus richness for that sample, and then we have number of samples. So this is essentially just a histogram with these different groups. And because later on we realized that because I didn't pair the pollen ball that I looked at with the mom who was foraging, it's not really statistically appropriate to compare between foragers and whole pollen balls, even if they're within the same year. So we ended up sort of just describing this data rather than looking for true significant differences. But it's interesting because you can see that the foragers tended to have similar numbers of genera as did the pollen balls. So this is seeming to suggest that they're foraging and choosing different plants within each foraging trip rather than, foraging for one thing on one trip, something else on the next one, etc. And I did have some data from 2021 as well, just foragers. And this is interesting because you can see that they tended to have a much higher richness across these samples. And I think this is because I collected these bees towards the end or the middle of the foraging season rather than the beginning. So this does give a bit more support to the hypothesis that shifting plant communities are allowing for different things to be collected, and in this case, many more plants to be collected. So we concluded that this is suggesting that these bees may mix and match across genera while foraging to balance pollen protein to lipid ratios. And here I have the data from the barcoding itself. Each of these individual lines, though it's hard to see, is an individual sample. And I have foragers from 2020, 2021, and then again, those whole pollen provisions. And along the side here, I have each of the plant genera that we detected. We didn't go to species level because this is quite difficult generally with this type of data. But I do have the proportion for each of these samples. So the darker the proportion, the higher the proportion of that genus within the sample. And you don't need to look at all of these different things. The important point I wanted to make is that most of these genera are not actually within the families Rosaceae and Fabaceae. They actually ventured quite widely outside of these two families. So it sort of brings into question whether or not they are mesolectic. And when we look at the proportion of host plant pollen, and I should say that these proportions are based on the number of reeds that we found during the sequencing run. And we've removed any reeds below, I think, 1%. So it's not going to detect pollen that would just appear by, a bee landing on a flower. And the foragers from 2020 only had a proportion of 0.12 host plant pollen within their pollen loads. Foragers from 2021, it was 0.31. And then the whole pollen provisions were a bit higher, with about half of them being made up of host plant pollen, but this is much lower than we would have initially expected. Okay, so the final question, do protein to lipid ratios found in the field correspond to the optimal diets for osmia during development? So to do this, I had a large walk-in chamber where I raised these bees in well plates with individual pollen diets that I made myself, and where I would transfer the eggs from the field to the new pollen diet. So for the first year, my experimental design was similar to the first year for that honeybee experiment. I had these altered pollen diets that I altered with canola oil and casein. I also included 2 control diets, one where I added the same amount of protein and lipid to sort of test the effects of the modifiers to see if they were toxic to the bees. And this was the same ratio as the unmodified control pollen diet. The following year, I looked at naturally different diets, again, similar to the last paper, ranging from very protein rich to very lipid rich. And then this time I also had a control diet where I took a bunch of osmia pollen provisions, homogenized them, and then made them into new balls and transferred the eggs to have a control that sort of tested for the egg transfer. And then I also that year decided to look at individual single plant species. So I had pollen from black walnut, ripwort plantain, black willow, orchard grass, and red maple. And then again, the same osmia pollen control. Okay, so jumping into the results here. Along the bottom, you have the diet and then just percent survival. So you'll notice that the control group did not perform significantly better than this diet where we added a bit of protein, but both performed better than the diet where we added a lot of protein. And the modified control also didn't do as well as the control, which seems to insinuate that these additives were not good for the bees. And this is further supported when you look at the very lipid-rich diet, which had no surviving bees, which technically isn't significantly different than these other groups, just because there were no surviving bees. But I think this means that the canola oil in particular was really harmful. So I was quite glad that we decided to do more than one field season with these different diets. But then when you look at adult survival, all of them did equally as well once they reached the cocoon spinning stage, if they survived the larval stage, except for the modified control with those additives. So here I'm going to present the results for the naturally different diets, which were honeybee pollens, and then as well as the single diets, which will pop up here in a bit. So the control group, the high lipid group, or excuse me, the high protein group, and sort of the mid-range diet did not perform significantly different from one another. But the 5-to-1 did perform significantly better than the very lipid-rich diet. And also compared to the 3-to-1 diet, which is interesting because this is the same ratio, or should be the same ratio as the homogenized control. So that seemed a little odd, and I think this could be because, well, I'll explain in a moment after I present the individual pollen results. Again, here, we're only comparing these diets with the control and these diets with the control, so these diets are not being compared to all of these over here. But we found the control performed better than all of these diets except for the Acer rubrum, which I think is the black walnut. I can't remember, but Acer rubrum. And this is interesting because these ratios are quite similar to what they should have been performing well on when you consider what they collected in the field. And the range in which from 0.75 to 1 all the way to 7 to 1, these diets should be okay, but they're not. And we think what's happening is that the plant species makeup may be more important than the overall macronutrient ratio, especially when you consider that pollen also has all these different micronutrients and secondary plant compounds that can be toxic. So perhaps the mix is more important than the nutrition alone. So the conclusions I drew from the study were that their target ratio was around 3 to 1, but the range was from 0.75 to 1 to 7 to 1, indicating a broader tolerance than the prior studies we looked at. We also concluded that those pollen provisions from individual foragers had a similar general richness to whole pollen provisions, which suggests that bees are mixing and matching or balancing pollen within each foraging trip. And I really think it would be cool if somebody followed up on this to see how exactly bees are making these decisions. Also, we determined that host plant families cannot necessarily be used to determine dietary tolerance because, again, the tolerance was much wider than what had originally been observed just for those 2 host families. And finally, pollen protein to lipid ratio is not the only measure of a suitable diet. Plant secondary metabolites and micronutrients are also influence diet suitability. Okay, so the final study, this is the one that isn't published just yet, that it's about Herites trincorum. So here I'm showing a graphic from Anthony Ayer's review that he conducted with Sandra Rehan in 2021. Excellent review. And it shows that urban areas tend to filter out bee species with particular traits. So it seems to favor generalist, small-bodied, late-emerging, and cavity nesting species overall. And this made me want to look more into how a specialist species would fare in an urban environment. We know that they wouldn't be favored, but I wanted to see if the overall proportion of urban area at a site would impact specialist bee foraging preference and nutrient acquisition. So if they don't have their host plant, are they going to collect from other host plants? And how does this affect the nutrition they're bringing in overall? So to give you some background, this study was conducted in Germany with the University of Freiburg with Dr. Alexander Klein and Dr. Felix Fernhoff. These bees are endemic to Germany and they're specialists on Asteraceae plants. And this is interesting too because Asteraceae are very common garden and weed plants, so there are quite a few native ones there as well. And we assumed that these weeds would be very common in urban environments. We thought this would make the question a little bit more interesting. Similarly to osmia, they nest in reeds, and they also make pollen balls and lay eggs in individual cells. My hypotheses for this study were that bees would produce fewer nesting cells in areas with increased urbanization due to fewer host plants and fewer native plants. Second, bees would collect more pollen from non-native and non-host plants in these urbanized areas when they couldn't access the preferred plants. And finally, that bees nesting in more urbanized areas would collect pollen with different nutritional compositions than those in less urbanized areas due to these changes in foraging behavior. So this is a map of all of our different sites across Germany. We sort of used impervious surface to determine how urbanized an area was. So we had sites ranging from low to high impervious surface. And I found these values by using GIS with a sort of a map of Europe that had this data. And I looked at 150-meter radius around these sites, and they are all considered independent. I know that these look pretty close, but when you zoom up on them, they aren't close enough for the bees to cross into the. So the variables of interest I looked at, first I had pollen diet composition variables. So this was again, we conducted metabarcoding for this study as well. So I looked at the proportion of barcoding reads from those genera. Sorry, my internet is apparently unstable, so hopefully okay. Can somebody confirm that I am still in the meeting?
Yes, you are still in the
Because it's
meeting.
saying, okay, everybody's still here.
Yeah, we can
Okay,
see
sorry, I
you and
looked
your
over and didn't see the Zoom box, so okay, anyway, thank you. Small panic. So again, we looked at the proportion of host plant genera and barcoding reads from host plants, same with native plant genera and barcoding reads. We also looked at the effective number of genera within these pollen provisions. So this is just a different measure of diversity similar to the Shannon index, but is a little bit easier to interpret. And then we also looked at the same dietary variables as before: protein, lipid, and protein to lipid ratio. I also, again, looked at the percent of impervious surface area within a site for this urban area variable. And then I also had two progeny health variables. I looked at the number of brood cells that were present within a nesting tube and the proportion of progeny that survived. Okay, so this was all done using generalized linear mixed models. So I'll show each of these plots and sort of explain the relationship that's going on here. So along the bottom is the proportion of impervious surface area. And on the Y are the number of brood cells per nesting tube. And this was a significant relationship. So the more impervious surface area, the less nesting, the less bees you have per nesting tube. And this was across 30 different sites with 163 different nests. And I calculated that for every 10% increase in impervious surface area, there were 0.2 less brood cells per nest, which doesn't sound like a lot, but when you think about it on a whole, when there might be 100 nests per site, that is quite a lot. And after I look at, after I present all the different graphs, I'll go through and show you the tally of the true impact. Here's the sequencing data. The main thing I want you to notice is that these bees did collect from a lot of non-host plants. So these are all the little blue asterisks, as well as non-native plants, which are in red. And we did find many new records for this species, which I thought was pretty cool, but I guess this species hasn't been studied in a while. So I'm excited to update those records. Yep, that is just the same. Okay, and then looking at pollen nutritional content, this is more of observation, not necessarily a testable hypothesis. So we got the distribution for the protein concentration, the lipid concentration that these bees were collecting, the pollen, as well as the protein to lipid ratio for these different pollen balls. And we did find that the target ratio was about 1 to 1, and it ranged from about 0.32 to 1 to 3.2 to 1. Though I will note that this was with only 20 nests at 17 sites, so these samples were much more limited than I would have liked. But what's cool is that they're collecting pollen similar or within a similar dietary range to that of Asteraceae, which ranges from 0.39 to 2.63 to 1, with a mean of about the same, about 1 to 1. And I did look for relationships between impervious surface, bee health, and protein to liver ratio that bees were collecting, and I found no relationships. Okay, so here I have all the different plots with the proportion of impervious surface area along the bottom, and all of these different variables along the Y. So first we have effective genus diversity within a pollen provision. And I do consider this relationship significant for this study because it's such a large ecological study. Oftentimes you can't go alone on using 0.05 as a cutoff for significance. And I know that this is a bit controversial, but I think it's important to look more at the overall relationship, how practically significant the result is, rather than just a strict cutoff point. So I did consider this relationship significant. And then all of these other variables, the proportion of host plant genera, native plant genera, host plant pollen, and native plant pollen, none of these were significant in their relationship to the proportion of impervious surface area. And then when you do the calculation for this relationship, it's a 5.8% increase in effective genus diversity for every 10% increase in impervious surface. Okay, so same thing here, but this time we have brood cells per nesting tube along the Y in the different variables across the x-axis. Starting again with effective genus diversity, we found a negative relationship between the number of bees per nesting tube and the number of genera that were present in a pollen tube, which seems counterintuitive, but I will explain in a bit what we think is happening. For the proportion of host plant genera, we found a positive result, as you might expect. And these other variables, the proportion of host plant pollen itself, so not host plant diversity, but pollen alone, and the proportion of native and native plant pollen were not significant. But something important to note too is that because this is an ecological study and we're just using what data we have, we didn't apply a treatment. You'll notice that these bees did manage to collect quite a bit of host plant pollen, so there isn't an equal distribution of the sort of treatment of lower amounts of host plant pollen. So it's kind of hard to assess these relationships, and I'm curious what you would find in a in vitro environment where you rear varieties on different native and host plants in a controlled setting. And so again, these are the calculations for those two relationships we considered significant. First, there's a loss of about 0.5 brood cells per nesting tube associated with every effective genus added to a pollen provision. And there was an increase in 0.2 brood cells per nesting tube with every 10% increase in the proportion of host plant genera. And so to make this a little easier to understand, for a site with 100 nests, you would have a loss of about 22 cells per 100 nests for every 10% increase in, oh, that's not right. I think this is supposed to be something else. I'm sorry. Okay. You would have a loss of 21 cells for every 10% increase in impervious surface area. And when impervious surface area ranges from 0 to 80%, that's the loss of quite a few progeny, which I think is quite significant. And then for a site with any number of nests, there would be a max increase in about half a genus for every 10% increase in impervious surface. So I think the highest diversity we recorded was 5 effective genera. So this is a little harder to say if this is truly affecting these bees. And then for the number of every genera added to a site, again, max of 5, you would have a max loss of about 3 brood cells for every genus added. Oh, and I should say that would be for an individual tube. So if you had 100 nests, it would be quite a few more. So the conclusions I drew from this study were that increased urbanization negatively impacts brood cell numbers for Herides truncorum. Increased urbanization results in a higher plant diversity, and it may lead to bees collecting pollen from less preferred resources due to a lack in host plants. So I think that this is why you would think that higher diversity would be good for the bees, but we think it's actually that higher host plant diversity is good and overall diversity is not good because it's less preferred resources. Nutrient acquisition and collection of non-native forage did not appear to be impacted by urbanization. So the bees didn't care if the plants were native or non-native, and nutrient acquisition did not change. And finally, we may be able to predict protein to lipid ratio requirements for a specialized species, like we did with Herides when comparing Asteraceae, their host plant family, with what they collected. Unlike in osmium cortifrons, where we were not able to do that because they were much more generalists than these bees. But further work would be need to be conducted to determine if this extends to micronutrients and if how this pattern would extend to other bees. And with that, I'd like to acknowledge my lab I came from before I was in Sandra's lab, the Grosinger lab, as well as all of my funding resources.